Previous year question which we need to study is which of the following is a polysaccharide? Which of the following is a polysaccharide? It is starch. Starch is a polysaccharide. This was asked in CBSE 2020. That is, the polysaccharide here present is starch. Now, what is the difference between native protein and denatured protein? Suppose imagine we have now native protein is something which has its own biological activity and it has its own unique 3D dimensional geometry. Now denatured protein is nothing but, now when this protein, suppose imagine a protein is subjected to some physical changes like change in temperature or pH, the hydrogen bonds in them gets disturbed. Now when it gets disturbed, the globulus present in them, it will get uncoiled, and then it will get uncoiled and what will happen, they will lose their biological activity. Now this, they lose their biological activity, and this is known as denatured protein. So first thing, once again we'll take it, that is when a protein is subjected to physical changes like temperature, change in temperature, change in pH, and if it's hydrogen bond, the nitrogen bonds will get disturbed and the globulus present in them, it will get uncoiled, and then what will happen, it loses its biological activity. This is called denatured protein. Native protein means you just have to write it's only 2 points, that is it has, native protein is it has its own, it is found in biological system and it has its own 3D dimensional structure and it has its biological activity. That is the difference between native protein and denatured protein. Now what is the protein which vitamin which is responsible for the coagulation of blood? Now what is the protein which is responsible for the coagulation of blood? It is actually vitamin K. So vitamin K, if you see, it is actually helpful for the coagulation of the blood. Next, here also one more thing you need to remember, that in native protein and denatured protein, in the denatured protein, right, 2 degree and 3 degree protein only will get changed, they'll get disturbed, whereas the other things, they will remain as same as it is. Now next question, that is, what do you mean by a nucleotide? A nucleotide is nothing but it is all 3 basic components of a nucleic acid, pentose, sugar, phosphoric acid. When a nucleoside is linked to the phosphoric acid at 5 degree position of sugar moiety, we get a nucleotide. So now we have a nucleotide. We need to know 3 things which it contains. It has 3 components. One is phosphoric acid, pentose sugar, and nitrogenous base. So PPN. So pentose sugar, phosphoric acid, nitrogenous base. It contains these 3 things. Now when a nucleotide is linked to the phosphoric acid, at which degree? At 5 degree positive with the sugar moiety, we get a nucleotide. Once again I'll repeat it. See, what are the important things you need to remember here? Nucleotide contains important 3 things, that it contains 3 important components. What are the 3 important components? The first one being pentose sugar, the second one being phosphoric acid, and the last one being the pentose sugar, phosphoric acid, and then the last one being the nitrogenous base. Now when the nucleoside is connected to a 5 degree into a phosphoric acid at 5 degree position of sugar moiety, that time we get a nucleotide. Now next is essential amino acids, anomers. Now this one I'll just explain, or you can go through the one which is already there in this definition also. Anomers, I'll just explain to you like that itself. See, anomers are nothing but they're the stereoisomers which differ in the orientation. Now what is anomers? Anomers are nothing but the stereoisomers, and they differ in the position of OH group at carbon first position. They differ in the orientation of OH group at the first carbon position. They are called anomers. You can also write an example as alpha glucose and beta glucose also. Now what I mean by essential amino acids? Essential amino acids nothing but they are the amino acids which are essential for the growth and development, and they are synthesized by the body, that are called as essential amino, that are not synthesized by the body. I'm sorry. So non-essential means they are required for growth and development, and they are not synthesized by the body, but this essential, this cannot be synthesized by the body. See, once again I'll repeat it. See, even I'm getting confused. Just remember, essential amino acids are nothing but these are the amino acids which are required for growth and development, and they cannot be synthesized by the body. That's it. These are the only things you need to remember. Example, you can take it as valine also. For this one, in this reference, most of the PYQs, like in this PYQ, they have asked only the essential amino acids. In most of the PYQ, they have asked difference between essential and non-essential amino acids. So you can write the difference as the first point as these are also required for growth and development. These are also required for growth and development. However, this cannot be synthesized by the human body. This can be synthesized by the human body. And we have an example here, which is we have like many examples like valine and stuff, valine, leucine, and all. Now, on the other hand, non-essential amino acids means like glycine, align, alanine, those things and all are non-essential amino acids. Now, deficiency which causes the rickets. We have two things here. Deficiency which causes rickets is vitamin D. Vitamin K, what does it cause? It causes the coagulation of blood. So now give an example of for each of the fibrous protein and globular protein. So we have already learned the difference between fibrous protein and globular protein. Fibrous protein means keratin. Keratin is also present in keratin and myosin. This is also present in hair also. So keratin and myosin and albumins and insulin. So deficiency which causes rickets is gonna be vitamin D. I'm sorry. So which causes rickets is vitamin D. This fibrous protein is myosin and keratin. Globular protein is albumin and insulin. Now write the product formed on the reaction of D-glucose. So just write CHOH the whole 4, CHO, CH2OH, D, this is D-glucose. When you add bromine water to it, what happens? This aldehyde here, it gets converted to acidic, it gets converted to acid. So CHO, CHOH the whole 4, again CH2OH, it becomes COOH, CHOH the whole 4. Again down you should write it as CH2OH. This is what happens. This is what happens when the D-glucose reacts with bromine water. And we also saw the example of fibrous protein and globular protein, where we learned that keratin and myosin is fibrous, globular protein is insulin and albumin is. Since it contains this is a glucose and it contains acid, we call this as gluconic acid. So there we saw how if suppose the glucose is like reacts with bromine, we get here instead of CHO, Now here what do we get? Here we get it as CHO, everything remains same. But the thing is, here CHO is there, right? It becomes CH double bond NH. So this NH will come and get attached here. So CHO, CHOH, CH2OH, here it comes what? CH double bond NH, NH2, NOH, sorry. C double bond O, NOH. So CHO, just remember to write instead of C, you need to write it as N and then you need to attach it to CH. So CHO, again CH, double bond NOH, and again CHOH the whole 4, again CH2OH. This is called as glucose oxime. Next we have something called as Zwitter ion. In Zwitter ion you can only draw this and this. It is it is enough. No, wait, I'm sorry. Draw this one. This is Zwitter ion. Now, what is exactly a Zwitter ion? It is like a dipole which contains both negative and the positive charge. They show amphoteric behavior because they react with both acid and bases. And also one more important point, the carboxylic group is it loses a proton. You can see it is losing a proton, and that is why it is negatively charged, and this is positively charged because it gives this hydrogen atom to here. So you can see in acidic medium, CO minus of this Zwitter ion accepts a proton to form the cation, whereas medium NH3 loses a proton to form the cation. Next is there are one more questions, that is, why cannot vitamin C be stored in our body? See, you see the vitamin C, right? It is soluble in our body, so it is basically it gets excreted in the form of urine and sweat and all, which I've also mentioned here. Next one, deficiency of vitamin which causes scurvy is vitamin C. Coagulation of blood is vitamin K. And then one more thing which we had learned was what? Scurvy is vitamin C. And we learned even something about vitamin D too, and that was rickets. Now, which deficiency causes scurvy? It is vitamin C. What type of linkage is peptide formed in the form of this thing is called peptide linkage. Next, write the product formed when glucose is treated with HI. So we know the formula for that one. That is nothing but C H O C H O H, the whole 4. Again we learned that it will come down as CH2OH. Now here instead of CHO, this thing it becomes N-hexane. That is what happens when it reacts with HI. You can see it. You can see it clearly now. Now that's it for the PYQs.